Hello everyone, this will be a guide for using Designer for RNAi interference-based pest control tool named dsRIP. And as you can see from our main page, we have 4 tools, and the typical pipeline starts with target gene selection. The RNAi target gene finder tool asks us to choose an order or species first of all. Here we have insect pest orders, mites and pathogens. Here we will go with coleopterans. And then we will need to choose a species within that order. And here we can go with Leptotarsa disseminata. And I will show you later how to work on any other pests, even though they are not listed above. Then we will need to select whether we want to use animal targets or pathogen targets. Our animal targets are coming from genome-wide RNAi screen in red flower beetle, and the pathogen targets are coming from few targets identified in fungi. Then there are also some other parameters. For instance, the first one is whether we should exclude targets with paralogs. This is important because paralogous genes can compensate for the function of the targeted gene, hence reducing the efficacy. So if you want to reduce that risk, you need to opt for this option. Also, dsRIP has the ability to filter target gene candidates based on minimum number of successful transfers to other pests. For instance, if we select it as 2 transfers, dsRIP will only recommend genes that worked on at least 2 pest species. Also, there is an optional area for filtering specific pathways such as proteasome and spliceosome. These were identified from genome-wide RNAi screen to be very effective pathways to target, but here we can leave this empty. Then when you are done, you can click on Find Target Genes, and immediately dsRIP will recommend us the RNAi target genes for our selected species. We have some details here, for instance the name of the gene in our selected pest species, the orthologous gene in tribolium, and its annotation here. And also on the right side we have the number indicating the number of transfers to other pests. So 5 means this particular gene orthologs have been transferred to at least 5 other species successfully. And then we can also click on this button to download full detailed results. Here you will see an Excel sheet containing the same information, as well as a FASTA file containing the gene sequences. Then when you are after this, you need to select for target gene candidates that you want to work on, and you can simply select them by clicking on them, and dsRIP will indicate the selected targets as highlighted here. Then when you are ready with your selection, you want to click on Design Double-Stranded RNA.